Developmental biologists joke that this is the most important thing that ever happens to you in your life, because we go from a sheet to a series of 3 cell layers and then into a series of tubes that form our actual mature body. And the first thing that happens is a little strip appears on the caudal end or the tail end of the developing embryo called the primitive streak. And at the very tip of that there's an area called the primitive pit and an important signaling area just ahead of that called the primitive node. This is the place where gastrulation happens, and it's very difficult to visualize this in 2 dimensions. So what we're going to do is take a little bit of Play-Doh here. We have the epiblast disc above, hypoblast disc below, but since that hypoblast doesn't really contribute to the mature embryo, we're going to peel that away for the moment and make a primitive streak right there. Now, what's going on at this point is the primitive streak isn't just a gap. The cells in this area are actively folding under. They're still dividing, and they kind of invade under the primitive streak and wash under to fill in the space that was beneath the epiblast and above the hypoblast. And in this process, they're actually going to obliterate all of the hypoblast cells. So we'll get rid of that. As they roll under, they're going to form a new layer, 2 new layers actually, that are being called the endoderm and the mesoderm. And as this process completes, what we're left with is this epiblast cell layer on top, which is now magically referred to as the ectoderm. In red, we have the mesoderm, and in yellow, we have the endoderm. Now, endoderm is not the same as the hypoblast. All these layers came from the epiblast cell. So, ectoderm in blue, mesoderm in red, and endoderm in yellow all come from the epiblast by way of gastrulation. Now, what's important at this point, we'll fish our abused epiblast back out here, is there's this group of cells on the midline of the epiblast that roll under through the primitive node, and on the midline of the epiblast are going to form a signaling structure that's incredibly important in embryonic development called the notochord. And it's just going to be a tube of cells sitting just under the epiblast, and it's going to be the structure that actually signals the formation of more important structures hereafter. And if we were to look for where that was located in this area, we would just have to slice and look for this nice purple dot. And this notochord may not do much in the adult, but it is going to signal the surrounding mesoderm and ectoderm to do a lot of impressive things and form the central nervous system and the epidermis, as well as all the connective tissue structures and muscles that are going to make up almost all the human body. And in yellow, these endoderm cells, what they're going to form is just the lining of the gastrointestinal, respiratory, and urogenital tracts. And we're going to follow that process in the next talk. Alright, now as we talked about before, we've gotten to the point of the trilaminar embryo, ectoderm, mesoderm, and endoderm, blue, red, yellow, all kind of controlled by the signals that are coming off the little purple notochord that's located right here. What's going to happen, one of the first and major events that the notochord signals, is an involution of the ectoderm to form the neural tube. And the neural tube is going to form all of the central nervous system. Now we're going to take this as an isolated piece of ectoderm to demonstrate what happens. The first thing that happens is just above the notochord, the ectoderm thickens and forms a little fold on the midline, and 2 protuberances right here. So we have a neural groove right on the midline, and then we have 2 neural folds on either side. These continue to approach one another until they actually zip together all the way down, leaving this little piece kind of hanging out in the mesoderm. So it's actually invaded the mesoderm. So what we'll do is just pinch those together, and this is happening all along the length of the developing embryo. And now it's time for one more cut, because that neural tube actually fully disassociates from the rest of the ectoderm, And the rest of the ectoderm seals itself back together up above, and this is going to form the epidermis thereafter. So a nice little sheet there, and below it we've got what you can squint and approximate to be a tube. And we have a better picture of that right here. So we'll take a nice little section of this right here and hopefully see the main thing I want to draw your attention to is this nice little blue tube right here. This is now the neural tube. It's going to form the brain, brainstem, spinal cord, all of the central nervous system. Just below it, in purple, is the notochord, which signaled it to pinch off from the overlying ectoderm. And now, we've gone from a nice red, homogeneous mesoderm to subdivided mesoderm. And this is subdivided because not only has neurulation been happening, but the notochord, and once the neural tube is in place, neural tube itself has signaled the mesoderm to further differentiate into a variety of structures. And what we have, in kind of nice pink here, is somatic mesoderm, which is going to form somites. Just past that we have the intermediate mesoderm in orange, that's going to form the gonads and the kidneys. And past that we have in pink and darker red what's called the lateral plate mesoderm. And the fact there's two colors there will imply that we're going to have a split in those two layers very shortly, and we'll talk about that in just one moment. So last time we talked about how, under the influence of the notochord, we formed the neural tube, the true ectoderm, which is going to form the dermis, and then endoderm below, and the segregation of the different parts of the mesoderm into the somatic, intermediate, and lateral plate mesoderm. Now, this is the point where we actually take these layers, these flat layers, and form a series of tubes. And if you want to look at the human body as a series of tubes, you wouldn't be far wrong. And the way this happens is that between two layers of this lateral plate mesoderm, we start to get little spaces that develop. And these little spaces are called intra-embryonic coelom, or cavities, and they get larger and larger until they really and truly separate the lateral plate mesoderm into two portions. The portion that's going to be in contact with this yellow section down here, the endoderm, this whole assemblage is now referred to as the visceral or splanchnic lateral plate mesoderm. And what it's going to do is fold on itself and create the gut tube and all the connective tissue and smooth muscle that surrounds the gut tube. So let's do that. And in order to